Welcome to the Route Lab tutorial on harvesting cells and making noodles. This protocol is designed to prepare yeast cell cultures for cryogenic lysing. These cryogenically lysed cells will later be used in protein isolation experiments. The number of cells you should grow is determined by the protein complex you are studying. In most cases, 6 liters of culture grown to a cellular density of 3.0 times 10 to the 7th haploid cells per milliliter, or an optical density of 0.8 will be sufficient. This will yield one 50 milliliter falcon tube worth of noodles. Once your cell cultures are at the appropriate density, spin them down using a high-capacity centrifuge. Here we are using 2 1-liter bottles to spin down our cells. Make sure each bottle has the same amount of culture to keep the centrifuge balanced. Using a high-capacity centrifuge, spin down the cell culture at 4000 G for 10 minutes at a temperature of 4 degrees centigrade. Keep your sample on ice at all times. For the purpose of this video, we did not put these bottles on ice. Once the culture has been spun down, you should have a large cell pellet at the bottom of your bottle. Discard the supernatant, being careful not to pour off any of your pellet. Repeat step 1 if necessary until all your culture has been spun down. Wash the cells by resuspending them in autoclaved, double-distilled water. 50 to 100 milliliters of water is usually sufficient, but more can always be used if needed. If you are working with just one strain of yeast, you can combine all of your pellets during this washing process. Transfer the resuspended solution to 50 milliliter falcon tubes. Spin down the solution at 2600 G for 5 minutes at 4 degrees centigrade. Repeat this wash and spin down step one more time. Discard the supernatant from the twice washed and spun cells. Then resuspend the cells with resuspension buffer, using a volume of buffer that is equivalent to the volume of your pellet. In this demo, our pellet was approximately 10 milliliters, and so 10 milliliters of buffer was used. Once resuspended, spin down the cells at 2600 G for 15 minutes at a temperature of 4 degrees centigrade. Discard the resuspension buffer and spin the cells a second time at 2600 G for 15 minutes at 4 degrees centigrade to ensure that all the buffer has been removed. Prepare your noodle making station during your last spin in a working hood. Place a 50 milliliter falcon tube rack into a styrofoam box and fill the box with liquid nitrogen. Then place an open 50 milliliter falcon tube into the rack and fill it with liquid nitrogen. Finally, place a sheet of aluminum foil over the rack to keep the liquid nitrogen from evaporating too quickly. After the final spin, the pellet should be fairly dry and resemble a thick paste. Using a spatula, transfer the pellet from the falcon tube to a 20 milliliter syringe. To make the noodles, press the cell pellet paste through the syringe and into the falcon tube with liquid nitrogen. When ejecting the paste from the syringe, try to do it in small spurts so the noodles do not form one big clump. Every once in a while, use a spatula to push already frozen noodles to the bottom of the falcon tube in order to make room for the rest of your sample. You can also use a spatula to help you break up the cell paste as it exits the syringe. Both of these methods will help you avoid having large clumps of noodles. Try to keep the falcon tube filled to the top with liquid nitrogen. Continue pushing the cell paste through the syringe until there's nothing left in the syringe. To decant the liquid nitrogen in the falcon tube, punch holes into the cap using an 18-gauge needle. Place the cap securely on the falcon tube and turn the tube upside down, allowing the liquid nitrogen to flow back into the styrofoam box. Your end product should resemble noodles. Store them in the falcon tube at -80 degrees centigrade until ready for use.